So I'll now hand over to Professor Fiona of Blackhall, who will introduce our next speaker.
Thank you very much. So it is my real pleasure to introduce Professor Phillip Monaghan, who I have known for some years at the Christie. He's a consultant clinical scientist, and he's been exceptionally committed to improving patient care, driving service development, and representing laboratory medicine nationally, internationally, sitting and chairing various groups in Europe and beyond. And I think his impact is best summed up in a little quote I found. He was asked why he entered laboratory medicine, and he replied that research performed in the lab translates directly to the clinic for improved patient care. The vast majority of clinical pathways are underpinned by high quality laboratory services, and we therefore have opportunity to influence and improve the quality of patient care and ultimately patient outcomes. And I think for undergraduate scientists, postgraduate scientists, a clinical scientist career trajectory with that potential to influence in the way that Phil has is perhaps not as well socialized. I know Phil has been heavily involved in training as an exceptional role model and passionate about building his speciality. I think with this chair conferred, we'll be able to continue to do that. So, Phil, without further ado, I will hand over to you to give us a little bit of a sense of what you have achieved over time.
Fiona, thank you very much for those kind words. It's an honour today to be presenting at the MAHSC Inaugural Clinical Chair Lecture Series. I'll just share my screen quickly. There we go. Can everybody see that?
Yes, we can.
Wonderful, thank you. So, yes, I was wondering what to speak about really. So I've given the title of my talk, Driving Discovery to Patient Benefit. I'll jump straight in terms of my career journey then. So I'm a local lad. I was born and raised in Stalybridge in Tameside, and I applied for a university place at what was then called UMIST in Manchester in 1999 in biochemistry with applied molecular biology. And I spent my first year on campus. The top left, you'll see the Wright Robinson Halls of Residence, where great first year, made lots of friends and memories. We actually had quite a good social life. We engaged in inter-floor football events, five-side tournaments, etc. Unfortunately, in my first year, I broke my arm at one such event. It was my writing hand, so I had to have a scribe for my first year examinations. But that was thankfully didn't end my university career at that stage. Did have the opportunity to walk past Archimedes having his eureka moment in the bath every morning, which was an inspiration. Also, during my undergraduate studies, I undertook some summer jobs, my first of which was at Manchester Royal Infirmary as a porter. And the chief porter gave me the job of delivering specimens to the clinical sciences building. So every day I'd travel with my biochemistry specimens to the auto lab, up to the clinical science building to drop off the virology, microbiology, and histopathology specimens. And I made some good friends and colleagues during my time as a porter at MFT. Steve Keeney in the genetics lab kindly also gave me a tour during my time during my summer job, which was great. I learned a lot, and it really gave me a key insight into the value of laboratory medicine. Also a keen follower of Manchester City Football Club, as is my family. And during the undergraduate studies in 2000 through to the 2002 season, I managed to secure a steward role at the then Main Road. This was before the glory days of now. I've given the old Manchester City badge here with the motto Superbia in Proelio, which means pride in battle. And we certainly needed resilience back in those days down in Division 2. I was luckily enough to qualify from UMIST with a first class honours in biochemistry and applied molecular biology, and it was during my third year undergraduate studies with Professor Lulian Lian looking at a redox enzyme, novel reductase 1, with some exposure to structural biology in her NMR nuclear magnetic resonance facility that Lulian and other colleagues encouraged me to reach out to Professor Nigel Scrutton, then at the University of Leicester, for some consideration of PhD opportunities. I took a PhD under Nigel's supervisorship. Nigel is shown in the top left here. In 2002, with co-supervision from Professor David Lays on the right, both eminent in their field of enzymology and structural biology. And this equipped me with numerous techniques in molecular biology, cloning, recombinant protein expression and purification. And after many attempts, we managed to crystallize the recombinant protein of interest, in this case, proline dehydrogenase, which you can see these nice yellow crystals in the bottom left here. We took these crystals in liquid nitrogen on a plane, believe it or not, to Grenoble in the south of France, to the European synchrotron radiation facility, a couple of PhD students and David, and we were able to use that facility to fire high energy X-ray beams at these crystals to generate X-ray diffraction paddles, and finally elucidate the X-ray crystal structure, three-dimensional structure of proline dehydrogenase, which we published in 2007. Nigel in 2005 took up a professorship post and a leadership role back at the University of Manchester, so his extensive research team, myself included, followed suit, and we for a time were temporarily put up in the Mill building in the top left here. This is back on the old UMIS campus and was a full circle for me. This is where I had much of my undergraduate studies, including my undergraduate research project on the 7th floor, so it was very familiar surroundings. And we eventually moved back into the new Manchester MIB, the Institute of Biotechnology, in 2006-2007. And it was during that time which, perhaps through some of my impatience, that I was struggling to see the direct application of the research I was conducting as part of my PhD and postdoctoral roles. And the college in the lab at the time signposted me to the NHS clinical scientist training program, which at the time was called Grade A, latterly more known now as the scientific training program, the STP. So I applied for the 2007 cohort, for which there were 5 places at the time, so I wasn't holding out too much hope, but I was very fortunate, very lucky to secure a place in the 2007 training cohort. My base hospital was assigned as Arrow Park on the Wirral, under the supervision of Dr. Maureen Leonard and Dr. Duncan Nethercutt, a consultant clinical biochemist and chemical pathologist respectively, both with an encyclopedic knowledge of clinical biochemistry, so I couldn't have been in a better place really. This included paediatric secondments at All Day Hospital, and also through my master's degree in clinical biochemistry, which I was undertaking at the time, I had my postgraduate research project at Wythenshawe Hospital, now part of MFT, under the supervision of Professor Brian Keeble, who is a world-leading expert in clinical mass spectrometry. I then gained my first substantive role as a qualified and state-registered clinical scientist in 2010 at the Christie Hospital in the path labs there. In 2014, the service went out to procurement, went out to tender, and there was a successful procurement by SimLab UK, and we were rebranded as the Christie Pathology Partnership. That was a 10-year contract, which was successfully renewed last year for a further 15 plus 5 years. Outside of the day job, I've been heavily involved with various opportunities, including with the European Federation of Laboratory Medicine, where I've been a member of the test evaluation working group since 2011, when I applied as a young scientist, back then when I was under 40. And this has been a huge opportunity to network and collaborate with colleagues throughout Europe and across the world. And this opportunity has taken me across Europe to various conferences and workshops and presenting opportunities. Also further afield, I visited Sydney in 2014 to provide a lecture series, attend conferences and workshops, and we even had a symposium at the IFCC World Lab Congress in Dubai in 2024. A huge thank you to Professor Rita Horvath and Professor Krista Cobart, who have chaired the Working Group for Test Evaluation, more latterly known as the Committee for Clinical and Analytical Performance Specifications, due to a recent EFLM restructuring. And I've been chair of that committee for the past 2 years, and I'm currently in my second term as chair. And we recently, back in May, had a specialist workshop looking at analytical performance specifications, specifically Model 1B indirect outcome measures for APSs, and this is the CCAPS committee and a broader field of experts. We're in the process of finalising a special issue publication in the journal Clinical Chemistry and Laboratory Medicine later this year. It was a very fruitful meeting. So I want to talk about some service developments in the Christie Pathology Partnership, where quality is all important in medical diagnostics laboratories. We're accredited to the by the United Kingdom Accreditations Service, UCAS, to international standards ISO 15189-2022. So science does not end where quality begins, and I just want to start with a couple of stats given by the Royal College of Pathologists. Around 95% of clinical pathways rely on patients having access to pathology services. 500 million and 130 million biochemistry and hematology tests respectively are carried out each year, and 50 million reports are sent to labs to GPs from labs every single year. So pathology and laboratory medicine is absolutely central and crucial to care pathways and patient care. The Christie itself was formally recognised as a centre of excellence by the European Neuroendocrine Tumour Society back in 2010. This is a molecular representation of a general neuroendocrine marker called chromogranin A. And we used to send this test away, but we repatriated it in 2011 when we conducted a clinical audit and found its utility over other markers such as chromogranin B. We started to analyse chromogranin A in-house on an enzyme-linked immunosorbent assay, or an ELISA, you can see on the top left plate here, which is very manual, laborious assay. And it was at a UKI NETS conference in 2012 that I bumped into a chap called Michael Trace, who was working with Thermo Fisher on a Brahms crypto automated assay for chromogranin A, and we managed to get hold of this platform in 2012 to transfer the assay, which was great. Better performance, higher throughput, more automation, more reliability. The problem with chromogranin A, it's a large heterophilic molecule, and different manufacturers and different assays, Cisbio, Dako, ED, Thermo, for example, use different antibodies to detect chromogranin, and the proprietary antibodies that they use detect different epitopes on different sites of the molecule. They report in different units, so it's completely unstandardized. So at the time, and I continue to work with UK NEQAS. I sit on the Immunology, Immunochemistry and Allergy Steering Committee with the Director of UK NEQAS, Dina Patel, shown here in the left. And I approached Dina to see if it would be worthwhile considering a pilot external quality assessment scheme for chromogranin. EQA, for those who are not aware, is a process whereby an EQA scheme distributes a sample to participating laboratories of unknown concentration, in this case for chromogranin. The laboratory would then measure that chromogranin and post the result back to UK NEQAS. UK NEQAS then compile a report, and it's a way to compare your performance, your laboratory's performance, against your peer groups and other laboratories for that analyte to assess performance, accuracy, precision, etc. Now, back in 2013, we had 5 laboratories enrolled using 3 different methods to detect chromogranin and reporting in 3 different units: nanograms per mil, nanomoles per litre, and international units per litre. Fast forward 13 years to 2016, this is the March 2026 distribution report in the bottom left corner here, and we had 134 labs involved and enrolled on this scheme, which is no longer a pilot but a full scheme. 104 of those are using the Cryptora platform. We're an inclusive scheme, so there are 11 methods in total, but what we've done is not standardize but harmonize the reporting units to a common reporting unit, a mass unit of nanograms per mil, which greatly facilitates labs to compare across different methodologies. And we've done this by utilizing the vast amount of EQA data we've got and formulating a conversion factor. And this will be hopefully being published fairly shortly. And just to emphasize the global reach of this EQA scheme, the 134 labs are dotted throughout the globe, so it's really had positive, meaningful impact worldwide. Moving on to research and innovation at the Christie, driving discovery to patient benefit. This is really where my passion resides. And within the laboratory and under the umbrella of research and innovation, the laboratory conducts day-to-day various aspects of research, be it assay verification and validation, post-market surveillance, determination and validation of reference ranges and reference intervals. We're sat on a wealth of real-world data in our laboratory information management systems and EPRs. So we're utilising that data now, not just for clinical audit, but now for developing predictive models using machine learning, which is coming to the fore. And we've published some data on that fairly recently. And we're now moving into the realms of biomarker research, formally funded biomarker research, and investigator-led clinical trials. The first example I want to give is from a study which I'm quite proud of. It was some 14 years ago now, but it was completely unfunded. And I wanted to look at cortisol measurement in patients with Cushing's syndrome receiving medical management by the drug metarapone. Now, Cushing's syndrome is the manifestation of pathological hypercortisolism, so cortisol excess from the adrenal glands, and that can be medically managed by metarapone, which is which blocks cortisol production from the adrenal glands. And Professor Peter Traynor, who until 2020 worked at the Christie, conducted some research on this back in 1993 and showed that endocrinologists, when they titrate the dose up of metarapone, should be aiming for a serum cortisol day curve therapeutic window, if you like, of between 150 and 300 nanomoles of cortisol circulating to achieve optimal response to metarapone. This is at the level of where the metarapone blockade is within the adrenal steroidogenic pathway. It's an 11-beta hydroxylase inhibitor. The result of that is you have a build-up of the cortisol precursors above that, upstream of that pharmaceutical blockade, and you can see they're all very structurally similar to cortisol, this consistent steroidogenic ring. That has implications for how we measure cortisol in the lab. If we take immunoassay, for example, it's a structural recognition assay whereby antibodies are used to bind to cortisol to measure the quantity in the blood or serum or plasma or urine or saliva. If you have structurally related steroid hormones floating around in that matrix, they are inevitably going to cross-react and cause falsely raised results due to that cross-reactivity. And indeed, that is what we see in immunoassay. Now, to look at this in more detail, I compared immunoassay. You can see on the left-hand side here, we've got a Centaur manufactured by Siemens, compared to a mass spectrometry assay for cortisol, which was developed at Wythenshawe Hospital in Brian Keevel's lab. We had a control group of patients where we saw a negligible bias here, 2.5%. A Cushing's group of 32 patients who were treatment naive, again, negligible bias, 1.3% bias between immunoassay and mass spectrometry measurements of cortisol. And then finally, a metarapone treated group of Cushing's patients where we saw a 23% positive bias on the cortisol when measured by immunoassay. So cortisol by immunoassay is effectively overestimating circulating cortisol for patients receiving metarapone therapy. This is just an example to illustrate it. We've had a recent patient at the Christie with Cushing's syndrome due to a pancreatic neuroendocrine tumour. The patient's dose was titrated from 750 mg 4 times a day to the maximum dose of 6 grams per day. And this graph just illustrates the difference. The top line is cortisol results by immunoassay. The bottom line is cortisol results by mass spectrometry. 2.3-fold difference in values. So the metarapone is having a direct impact on the measurement of cortisol, which can have implications in terms of appropriate clinical management decisions in that regard. So we published this data in the Annals of Clinical Biochemistry back in 2011. The main points and main implications were the potential for overtreatment with metarapone. The endocrinologist can see that there's a high result from an immunoassay-based cortisol and continue to titrate up that metarapone. That can cause a significant risk of unrecognized hypoadrenalism. The recommendation from this paper was to use LC-MS or liquid chromatography tandem mass spectrometry, mass spec for short, in patients receiving active metalloprone therapy. I've just shown Professor Peter Traynor, top right here, Professor Brian Keeble, and Laura Owen, who are instrumental in this study. And a big thank you to Peter Traynor, who's been a huge mentor for me throughout my time at the Christie, and really fostered my interest in biochemical endocrinology. This paper found its way into manufacturer kit inserts. So in instructions for use, this is the Siemens Healthineers cortisol kit insert as an example. This is now provided as a limitation of the assay. Before it was a relative unknown. Furthermore, and this was quite a proud moment for me, I saw that my paper had been cited in the US guidance, the Endocrine Society Clinical Practice Guideline for Treatment of Cushing's Syndrome, by Lynette Nyman et al., which advocates the use of mass spectrometry for patients being treated with these 11-beta hydroxylase inhibitors. I was fortunate in 2012 to be awarded by the Royal College of Pathologists the Specialty Research Medal for this paper, and I was presented the medal award by the then president, Dr. Archie Prentice. You can see I still need to learn how to smile on photos. Now, moving on to biomarker research and funded studies. This is a current open trial called VALTIVE-1, which is the validation of plasma Tie2 as the first vascular response biomarker of VEGF inhibitors, in this case bevacizumab. Ovarian cancer stats are quite stark from the Cancer Research UK site. 21 new cases per day, 3,900 deaths per year, so there's a clear unmet need here. Now, the CR UK funded trial is a £1 million grant to develop this novel biomarker and validate it for clinical use. £250,000 of which came to my laboratory to validate the assay to NHS standards, and that is to ISO 15189-2022. There's a clear unmet need for bevacizumab guided therapy to improve utility and cost effectiveness of this therapy. And through the work of Professor Gordon Jason and colleagues, Gordon shown here on the left, Tie2 has been shown as a novel vascular response biomarker for anti-angiogenesis treatments. Tricon-7, Travastin, ABC3 are all published in ovarian, colorectal, and biliary tract cancers respectively. The Capilia trial in endometrial cancer has been submitted for publication. ICON-9 in ovarian will be a data readout shortly. The active VALTIVE-1 trial in my laboratory is due to close later this year, and including completion of data analysis. And we're about to start VALTIVE-C in colorectal cancer, for which we've just recently received the grant award letter. So we're creating a solid body of evidence here to evidence the utility of Tie2 for clinical use in the NHS. We've published a protocol in BMC cancer, and the previous work of Gordon and colleagues has shown that Tie2 has a vascular response of greater than 25% after 9 weeks on VEGF inhibitor therapy. So the aim of VALTIVE-1 was to really optimize that vascular response signature and validate it for clinical use in the NHS. And this is the CR UK Manchester Institute roadmap. It's a 2-stage qualification process, moving from GCP to ISO 15189 accreditation in my laboratory. VALTIVE-1 will define that vascular signature and optimize it, and power the subsequent randomized discontinuation trial, which is VALTIVE-2, and that will complete the roadmap for implementation, translational implementation into the NHS, which is a huge undertaking. It was useful to see the CR UK Clinical Research Committee feedback from the VALTIVE-1 study. The committee acknowledged the value of having the assay equipment at the Christie Pathology Laboratories as a CR UK resource. So I opened the Translational Biomarker Unit at the Christie Pathology Partnership back in 2020 off the back of this funding from CR UK, and we are the lead lab to translate assays into NHS standards for CR UK now moving forward, which is huge. Some practical points here in terms of VALTIVE-1 data pending. We've evidenced that the stability of Tie2 in plasma is stable at ambient temperatures for at least 3 days. Now that has significant positive implications clinically and operationally. So VALTIVE-1 being a multi-center study, it enables trial sites to just post samples through the standard post rather than have to worry about freezing and arranging couriers for these. So much more efficient, much more cost efficient from a logistical perspective. Reproducibility of on-treatment Tie2 changes is wholly important for the concept here. So we looked at comparing the Octopus trial with patients on paclitaxel therapy against the ICON 7 trial with patients on carboplatin and paclitaxel. We saw a 6% and 4% increase respectively. So changes in Tie2 were equivalent, and importantly, cytotoxic chemotherapy does not impact on plasma Tie2 concentrations. Similarly, we looked at the first 45 patients of the active VALTIVE-1 study, patients receiving bevacizumab, and compared that to the bevacizumab treated ICON 7 arm of the ICON 7 trial, and we saw a 12.3% and 11.8% reduction respectively from those two trials with patients receiving Bev. So the two studies showed equivalent Tie2 changes following VEGF inhibitor therapy on bevacizumab, which again is really positive news. And we also found in the VALTIVE-1 trial from the first 45 patients enrolled a 28.9% non-response rate, which was consistent with our hypothesized 25% non-response rate. So the data is looking very positive on that front. Now, looking towards the future, what does success look like? Well, subject to the VALTIVE-1 trial, we want to establish ISO 15189 accreditation for this new assay, scale this up in the clinical setting and provide a national service, and we're in discussions with manufacturers at present. This will hopefully lead to further increased funding, an effective translational biomarker pipeline, of which Tie2 and VALTIVE will be a key exemplar, and ongoing formulation and fostering of networks of clinicians, academics, and industry partners for the future. Importantly, for the new Christie Pathology joint venture, we'll be moving into a new facility in 2027. This is currently being built on the Christie campus, so we're growing and expanding our footprint for biomarker research and innovation for the future. And that will also be underpinned by a new innovation accelerator fund, which we're developing to improve research and innovation for pathology at the Christie for Christie patients and beyond. And finally, to my acknowledgements, which are extensive, and I hope I've not missed anyone, but first of all, to the VALTIVE Trial Management Group, particularly Gord and Jason as Chief Investigator, and likewise Mike Braun as Chief Investigator for VALTIVE-C, which will be opening soon, and the rest of the team. The Translational Biomarker Unit at the Christie Pathology Partnership, the Christie Endocrine team, which Peter has been absolutely instrumental in my career development, the Clinical Outcomes Data Unit, and liaising and collaborating now with Lauren Scanlon on some AI and machine learning opportunities. Martha Corbinitz at the Queen Mary University of London, the whole committee of the Clinical Analytical Performance Specifications at the EFLM, particularly Rita and Chris, who I've mentioned, Ravi and Pinaki at the Chandigarh Centre in Northern India, who I've collaborated recently on with IGF-1 and IGF-BP3 reference interval determination large-scale study for pediatrics and adults in Northern India, which improves accessibility of healthcare and remedies some healthcare inequalities for underrepresented groups. And finally, to Martin Bidlingmeyer at the Endocrine Centre at the University of Munich, who we're having ongoing collaborations with regards to long-term stability of IGF-1 lot-to-lot changes. And finally, to my family, my son Jacob, my wife Jenny, and my daughter Betty, who have supported me all through my career development. They have to put up with me on a daily basis, and they certainly keep me in my place. So I love them, and thank you very much. And not forgetting my Cavalier poodle Jessie, who takes any opportunity to have a sunbathe. And with that, I'll say research is for everyone. Be curious and collaborate widely. Thank you.
Thank you so much, Phil. It's fantastic to see that trip down memory lane as well, and that it all began with you portering those samples around. I think you must have received and possibly portered many of mine with from my clinical work over time. So I think a really clear illustration of the importance of the speciality. And so maybe if I can steal the first question around that pipeline of clinical scientists, the training routes, is it healthy or is it a neglected tribe in NHS careers, and what's the situation there?
My personal viewpoint is it's very healthy in terms of the infrastructure. It's provided through the University of Manchester, and my colleagues Helen Jopling, Phil MacDonald, they're doing a tremendous job there. There's still a very high application rate there for what is a relatively small number of opportunities through the National School of Healthcare Sciences. It's important that we foster these early career clinical scientists and research scientists moving forward, and particularly I feel quite strongly about ensuring that time is given for research, not just during their formal training, early training years, but then onto their first substantive posts, to reach out to clinical teams, look at where there is unmet need, identify opportunities within the laboratory, and ideally ring fence time to conduct research. I don't think that's happening across all areas of the NHS at the moment. There are some really good examples across the region here, particularly in Manchester, but that is something that we really need to keep a close monitor on for now and in the future, in terms of developing that workforce, not least as part of the NHS 10-year plan and the workforce plan which underpins that, because there are changing models of care, and pathology and laboratory medicine is really central to that, not just in the laboratory but out in the community. So I can see the roles of clinical scientists being more diverse and changing in the future, and that is where research and innovation will really come to the fore.
Thank you.
Jonathan.
Yeah, I think probably just following on that. So hi, Philip. I'm Programme Director for the Academic Health Science Centre at Health Innovation Manchester. And I suppose one of the, where we'll be focusing for the future of the Academic Health Science Centre is in, providing those research opportunities for all. And I think hearing your story, and congratulations on the chair, by the way. I think, we should, kind of include you as we move forward with those plans. I think it'd be really good to kind of get your insight there. So thank you for presenting today.
Thank you, Jonathan. Yes, very happy to contribute. Thank you.